Have you ever stopped to think about what makes the liquid medicines we rely on totally safe? We're about to pull back the curtain on an incredible, hidden process that's absolutely essential to modern medicine. It's the unseen science that keeps us all safe. Let's dive in.
So, it all starts with this really fundamental question. You know how we sterilize things, right? We usually blast them with heat in an autoclave. It's simple. It works. But what do you do when the very thing you're trying to protect would be completely destroyed by that heat?
And that is a huge problem for so many of today's most advanced drugs, especially things like biologics. If you hit those with high heat, you'd literally just cook them, making them totally useless. So you need a different strategy. You need a guardian that can get rid of all the dangerous microbes without harming the medicine itself. And that, my friends, is where sterile filtration comes in.
So let's meet the hero of our story, a seriously high-tech piece of equipment called the sterilizing grade filter. And trust me, we are not talking about your morning coffee filter here. Not even close.
Okay, so what is it? A sterilizing grade filter is this incredibly precise membrane. Its pores are just 0.22 micrometers or even smaller. Now, to put that into perspective for you, a single strand of human hair is about 70 micrometers wide. That means these pores are over 300 times smaller. They're so tiny, they can physically block bacteria and other microorganisms from getting through. It's like a bouncer at the world's most exclusive nightclub. So how good does it have to be? Well, to earn that sterilizing grade title, a filter has to prove it can stop a massive bacterial invasion. We're talking about a challenge of at least 10 million bacteria for every single square centimeter of filter space. And it has to stop all of them, letting only the sterile liquid through. That's an absolutely incredible performance standard.
But before a filter even gets a chance to prove itself with a real drug, it has to go through this super intense background check. It's a process called qualification, and it's all about making sure the filter itself is top-notch.
And this whole qualification process really boils down to two big questions. First, is the filter itself actually safe and pure? And second, is it tough enough to handle the real-world conditions of making drugs? Let's break that down.
For safety, the first rule is do no harm. The filter can't add any new problems to the mix. It has to be non-toxic, and it can't have any pyrogens. That's nasty stuff that can cause a fever if it gets in your bloodstream. And, critically, it can't have extractables. That's just a fancy word for tiny bits of the filter material itself flaking off into the medicine. You definitely don't want that.
Then you've got performance. Can it actually do the job? Can it handle a massive volume of liquid, the throughput, without getting all clogged up? Can it keep up with the speed of manufacturing? Can it survive the intense pressure and temperature, not to mention being sterilized itself with super hot steam? And maybe the most important question of all, is it compatible with the drug, or will the medicine just cause it to fall apart?
Okay, so once a filter passes that general background check, it's time for the main event, validation. This is where a specific filter is tested with a specific drug under the exact conditions of the manufacturing process. This isn't some generic test, it's a completely custom-built trial.
And the goal here? It's simple, and it is absolutely non-negotiable. The whole point is to prove that this filter, with this drug, under these conditions, will produce a sterile product. Not just once, every single time. There's zero room for error. So to run the ultimate test, you need an ultimate opponent, right? Scientists use this one particular bacterium called Brevundimonas diminuta. Why this one? Because it is famously tiny and known for being able to wiggle its way through the tightest spots. It's basically the Houdini of the microbial world, which makes it the perfect worst-case scenario for testing a filter. And this is where things get really fascinating. It's a real scientific puzzle. You need to challenge the filter with live bacteria to see if it can stop them. But what if your drug is, say, an antibiotic or has preservatives? It might kill the bacteria before they even get to the filter. So, how do you test the filter's stopping power then? So, the very first step is to answer one critical question. Can our little escape artist, B. diminuta, actually survive in the drug product? The answer, a simple yes or no, sends the entire validation test down one of two completely different paths. It's a real fork in the road. Okay, path number one. This is easy street. If the answer is yes, the bacteria can survive in the drug just fine, well then the test is pretty straightforward. You just add those 10 million bacteria per square centimeter directly into the drug product and run it through the filter. It's the most direct, most realistic test you can possibly do.
What? If the answer is no, and the drug is a bacteria killer, that's when scientists really have to get creative. They've got a whole bag of tricks. They might tweak the process, maybe by lowering the temperature. Or they could use a surrogate fluid, basically a placebo that acts just like the drug but isn't toxic. Sometimes they might even have to slightly change the drug's recipe just for the test, or pick a different, tougher bacteria that can actually survive.
But hold on. Validation isn't just about what the filter takes out. It's a two-way street. You also have to worry about what the filter might accidentally add to the drug, or even what it might unintentionally remove. And this brings us to two sides of the same coin. We already talked about extractables, making sure bits of the filter don't end up in the medicine. But on the flip side, you've got adsorption. This is where the filter material itself acts like a magnet and pulls the good stuff, the active ingredient, right out of the drug. Both outcomes are bad, and both have to be tested for and prevented.
Now, you might be thinking, do they really have to go through this whole complex process for every single strength of a drug? And thankfully, the answer is usually no. They use a smart strategy called bracketing. They'll test the filter with the highest and lowest concentrations of a drug. The logic is, if it works perfectly for both of the extremes, you can be confident it's going to work for everything in the middle.
So, with such a high-stakes process, the question of who's responsible is a big deal. I mean, where does the buck stop? Who's ultimately on the hook to make sure all of this works perfectly?
Well, the roles are actually very clearly defined. The company that makes the filter is responsible for all that general qualification stuff, proving they've made a safe, high-quality tool. But it's the drug company, the user, that's responsible for the product-specific validation. They have to prove that the filter works for their drug. And you can see by that double check mark, the final ultimate responsibility rests squarely with them.
And that really is the bottom line here. While the filter maker provides the tool, the pharmaceutical company that puts its name on that final medicine is 100% responsible for making sure the whole system is validated and used correctly. There are no excuses and absolutely no passing the buck. So, from a tiny bacterial escape artist to this intricate dance of chemistry and engineering, sterile filtration is this whole hidden world of science and responsibility. It all happens behind the scenes just to guarantee that patients are safe. So maybe the next time you see an IV bag or a vial of medicine, you'll think about that invisible, microscopic guardian standing watch, making sure what's inside is perfectly pure and safe.